Okay, now that we know more about the plasma membrane and what all that it's made out of, and the fact that its job is to regulate what can go in and what can come out by being selectively permeable, let's learn about the ways that we move this stuff across the membrane. There are two types of transport across the membrane. Today we're going to study passive transport. Passive transport does not require energy. That's why it's called passive. Think about when you're going on a long road trip. If you're the passenger, you can sit in the back seat and sleep. You don't have to pay attention to what's going on. You can just relax. That's the same idea with passive transport. The cell gets to act like a passenger. It doesn't really have to pay attention to what's going on because it doesn't have to expend the energy. It can just sit there and sleep, and this stuff is going to happen. Okay? So that's the most important thing to remember about passive transport. It does not require energy. Okay? And then just a reminder, what is the energy molecule called? ATP. And what organelle makes that energy again? The powerhouse of the cell? The mitochondria. Okay, I'm trying to show you guys these connections. When I ask these questions in these videos, you should be trying to think of the answers in your head. Okay, keep yourself studying, keep yourself going. The second most important thing to remember about passive transport is that molecules are going to move from an area of high concentration to an area of low concentration. Okay, so what's that mean? It means that if there's an area on one side of the membrane where there's a whole bunch of that molecule and they're all crammed together, they want to spread out. Okay, remember, everything wants to reach homeostasis. It wants to reach an equal level. Okay, think about if for some reason they put 200 students in this classroom. Okay, it's a big classroom, but it's not that big. So if 200 of you are in that classroom and I open the door, what are you guys going to want to do? You're going to want to go out in the hallway so that you can spread out so that you're not on top of each other. Okay, so you're wanting to move from an area of high concentration of students, 200 students in a classroom, to an area of low concentration. There's only 2 or 3 students out in the hallway. Okay, same idea. These molecules want to spread out. They want to reach equilibrium, high to low. So there are 3 types of passive transport. Okay, all 3 types require no energy. All 3 types are going to have molecules going from high concentration to low concentration. So type 1 is called simple diffusion, or sometimes it's just called diffusion. Okay, same thing, 2 slightly different words. So who can do simple diffusion? Simple diffusion is going to be really, really tiny, tiny particles. Good examples are gases like oxygen and carbon dioxide. With simple diffusion, these molecules are going to go right through the plasma membrane. They're going to go right through that phospholipid bilayer. Okay, very simple, doesn't require any help, doesn't require any energy, just goes. Okay, and as you can see in the image down here, this dotted line represents the semi-permeable membrane or the cell membrane. And on one side, we have a whole bunch of this really teeny tiny molecule that is able to do simple diffusion. On the other side, we don't have anything at all. Okay, so what's going to happen is all these little molecules over here, they're all crammed up, they're uncomfortable, they don't have any space. They're going to want to spread out. Okay, so that's what's going to happen. These molecules are small enough, they're going to go right through the membrane and start to spread out. And they're going to continue to move through the membrane until they are at equilibrium. They are equal molecules on both sides of that semi-permeable membrane. So now they're a little bit more comfortable because they've got more space, more room. Now at this point, they're going to start to mingle. Every once in a while, just by random chance, one of these molecules is going to move on over to this other side, and same thing is going to happen here. One of these is randomly going to move on to the other side. Okay, so they're still moving, but they're going to stay in that equilibrium. They're not going to bunch up all on one side anymore. You can do an example of simple diffusion at home all by yourself. You can just take a glass of water and drip some food coloring in it. Don't stir, because what's going to happen is those dye molecules are going to want to spread out into the jar. And so eventually your glass is going to be all the exact same color, even though you didn't stir it. That's an example of simple diffusion. Our second type of passive transport is called facilitated diffusion. These molecules are going to be a little bit bigger, so they can't just go right through the plasma membrane. They need some help. They need a facilitator. That's what a facilitator is. It's someone that just helps you out. So facilitated diffusion is going to require one of those transport proteins that we talked about inside the plasma membrane. The proteins that are going to be used are those ones that are always open. They just have a small opening, and the exactly right shaped molecule can just pass right on through. Again, since it's always open, it's not going to change its shape. It's just going to always remain that open shape. It doesn't require any energy, and these molecules are going to go from an area of high concentration where they're all cramped up, passing through into an area of low concentration where they can spread out and reach equilibrium. So just to repeat one more time, no energy, moving from high to low, as you can see in both of these images. Simple diffusion, really, really tiny, goes right through the plasma membrane like we see here. Okay, facilitated diffusion, particles are a little bit bigger, so they need a little bit of help. They need to be facilitated by using one of these always open transport proteins. Our third and final type of passive transport is known as osmosis. Osmosis is a special word that is, it means the diffusion of water, okay? Only water can do osmosis. Nothing else can do osmosis. Only water, okay? Now what this is where water is going to move through the semi-permeable membrane, and everything else is really big, so it stays put. So this is where water is going to move to reach an equilibrium of water. So again, you're going to go high concentration of water to a low concentration of water. So in this image here, we have the original U tube, where at the bottom we have a semi-permeable membrane. So this is going to act like the plasma membrane. On one side, we're going to put a high concentration of water with a little bit of solute, something that is too big to move through the plasma membrane. On this side, we're going to have a little bit of water and a whole bunch of solute. So what happens is the solute, we're not in equilibrium. There's a whole bunch over here, a little bit over here, and water can move, but the solute can't. So what's going to happen is the water is going to move. So you see here we have equal levels of water, but on the other side, since the solute couldn't move, those particles couldn't move, the water moved. Okay? The water moved over, and so now the concentrations are equal. Okay? Equal amounts of water per molecule. Okay? So this is the movement of water only. Now, osmosis comes with some new vocabulary words. These words are all on your word wall, but we're going to go over them together here as well. Okay? I'm going to go ahead and start talking about these. Pause to copy this chart if you need to, if I'm going too fast. Okay, so we've got three terms: hypertonic, hypotonic, and isotonic. These words are used to describe the environment. Okay, that is the most important thing to remember. The proper usage of these terms is in relation to the environment. The environment is hypertonic. Okay, so hyper means more than. So a hypertonic solution has more solute in the environment than in the cell. What happens is, since there's a whole bunch of solute outside and only a little inside, the water is going to rush out of the cell, and the cell is going to shrink, and it's going to wrinkle and shrivel up. Okay? The way I remember this one is that when I was a little kid and I was hyper, my mom would kick me out of the house and tell me to go play outside. So that's what happens in a hypertonic solution. It's hyper, so the water gets kicked out of the cell and it has to go outside and play. Okay, that's how I remember it. Hypo means less than. So in a hypotonic solution, there is less solute in the environment than in the cell. Less in the environment than in the cell. So the opposite is going to happen. Water is going to rush into the cell. The cell is going to swell up and get big and fat. And if it's an animal cell, because animal cells are soft and squishy, it might lyse, meaning it might burst open and die. Isotonic, remember our prefix iso means the same. So in an isotonic solution, the environment has the same amount of solute as the cell. So they are equal. So what's going to happen is some water is going to move in, but at the exact same time, some water is going to move out. And so the cell is going to stay the same. It's already in equilibrium. It's happy. Here's a picture of what happens to blood cells. In isotonic situation, the cells are happy. Water moves in and out at the same rate. They kind of look like little donuts. Put it in a hypertonic solution. All the water gets kicked out of the cell, and it shrivels up and gets wrinkly. Doesn't even look like a blood cell anymore. Hypotonic, water rushes in, the cell swells up. See, it's supposed to have this little divot in the center to make it look like a donut, but it doesn't because it's all swelled up. Now, these are examples of animal cells because animal cells have just a plasma membrane. They're soft and squishy. But let's remember, plant cells are different. Plant cells have an extra layer, that cell wall that makes them hard and rigid. So there's some special words for what happens to plant cells. If you put a plant cell in a hypotonic solution, remember hypo means there's less in the environment, water rushes in. With an animal cell, the cell might burst open from it swelling up, but plant cells can't burst because they have that cell wall which is protecting them, making it hard and rigid. So what happens is the vacuole fills up with water, and as it continues to suck up water, it presses harder and harder and harder on the cell wall, and it builds up pressure. That pressure building up on the cell wall is called turgor pressure. Okay? When a plant has turgor pressure, it's very rigid, and the plant's standing straight up, and it's a pretty happy plant. But what happens if you put it in a hypertonic environment? Hyper, the water gets kicked out of the cell, so the vacuole shrinks because it's losing water, but the cell doesn't shrink because of that cell wall. So it stays a little bit rigid. This is called plasmolysis because what's happening is the plasma membrane pulls off of the cell wall. Cell wall stays put, but the plasma membrane shrinks in. Plasmolysis, the loosening of the plasma membrane, okay, if you're using your prefixes and suffixes. Okay? Now, what happens here is since we've lost that pressure from losing all that water, is the plant's going to wilt, and it's going to look like a sad, wilting little plant. Okay? So make sure you understand these terms, turgor pressure and plasmolysis, and you can use them appropriately. Super study the terms hypo, hyper, and isotonic. Those three are really important, and they're really confusing. So you need to work with them to make sure you know when to use the right one at the right time. And remember, hyper, hypo, and iso refer to the environment, not the cell.